So, students of BT third sem, today I am going to discuss with you all about the RNA editing. Um, so let's get started now. Now, this RNA editing is actually is given in your genetics syllabus. So it is a part of molecular biology. But here, as alteration of bases are being done, which leads to modification of the proteins. Okay, just like alternative splicing, it is also a part of post-transcriptional modification before that particular messenger RNA is being translated into protein. So let's get started now. So about the RNA editing, what it says, let me explain. So RNA editing is another mechanism that allows an RNA to be changed after transcription. So the sequence of the RNA gets changed by RNA editing also, so as to encode a different protein from that encoded by that same gene. So just like we have said I have said about alternative splicing, that in that process we make different combinations to remove out the introns and exons. We skip the exons, okay, by different mechanisms, as I have said in the previous video. In RNA editing, we are actually altering the sequence of the RNA to be changed after transcription, so that that particular RNA will encode a different protein from that same gene. Okay, so it's a simple process through which it is actually a molecular process through which some cells make distinct changes to the specific nucleotide sequence within the mRNA, within the rRNA, also tRNA molecule, after it has been generated by RNA polymerase. Okay. Now there are two mechanisms for RNA editing. Basically, one is your enzymatic modification. That means with the help of enzymes, we will modify the bases. We will modify the nitrogenous bases. From one purine form, we will make another purine, or from one pyrimidine form, we will make another pyrimidine, or we can also convert one purine into one pyrimidine by different enzymes. We all know, like deamination, where we have studied that in mutation chapter, in deaminase enzyme is responsible for um, the deamination process, where we can convert a nucleotide into another nucleotide, another nitrogenous base, better to say. So enzymatic modification of the bases, so the nitrogenous base can be modified with the help of enzyme that are generating forms that alters how they are read by the tRNA. So if we modify the nucleotide sequences within the RNA, then within the mRNA, then automatically that base sequence will now code for a different codon that will be coding by different amino acids. Okay, like for example, deamination of bases like cytosine. Here it is saying site specific deamination of the bases like cytosine or adenosine by the help of the enzyme called deaminase, that cause change in the nitrogenous bases, that result in the change in the codons. It result in the change of the codons that are read by the tRNA to code for a different amino acid. So the first type of RNA editing is with the help of enzyme we are modifying the nitrogenous bases like cytosine, like adenosine. It is modified with the help of an enzyme called deaminase enzyme that cause change in the bases and result in the change in the codon sequence also, which will be read by different tRNA to code for the different amino acid, right? Like AUG. AUG is a start codon, right? AUG is a start codon, or better to say, I am giving another example. Suppose, um, suppose UUU. Okay, UUU code for a different amino acid, right? Now, suppose due to it is deaminated. Okay, sorry for that. Not UUU. It will be suppose cytosine CCC. It codes for a different amino acid. Okay. Now, suppose deamination of cytosine occurs, and suppose it converts into that cytosine converts into, due to deamination, it converts into what? It converts into, suppose, uracil. Okay, due to deamination of cytosine it converts into uracil. Then now becomes UCC. This will be coding for a different amino acid, right? So in this way, due to enzymatic modification by the due to enzymatic modification of the nitrogenous bases, we are actually generating different forms of the codons, which will be coding for a different amino acid. Now, another involvement is insertion or deletion editing. Okay, so the first example that we have seen is is base modification with the help of enzymes. Okay, like for example, in base modification, I have said that when cytosine gets deaminated, it converts into uracil. Okay, then another enzyme is there, adenosine deaminase, which converts the adenine into inosine. Okay, in adenine into inosine. So this type of enzymatic modification can be involved for RNA editing. The second mechanism that that can be applied in RNA editing is insertion or deletion editing. In insertion or deletion editing, we are actually inserting multiple nucleotides within the mRNA, so that there will be shift in the or change in the codon sequence, okay, within the open reading frame, like insertion or deletion of multiple uridine nucleotides within the mRNA at specific sites with the help of a guide RNA, and some of the enzymes like endonuclease, ligase, we can modify the mRNA. So insertion or deletion of nucleotides, multiple nucleotides like uridine within the message, within the mRNA at specific sites with the help of enzymes, and also with the help of guide RNA. Okay, with the help of guide RNA. Guide RNA are actually specific RNA sequences. Guide RNA are specific RNA sequences that function as guide for RNA, or they function for to guide the RNA targeting enzymes or DNA targeting enzymes which they form complex with. Actually, guide RNA actually complex with the enzymes, okay, to actually complementary base pair with the target site, and that enzyme will catalyze the reaction actually. So insertion or deletion of multiple uridine nucleotides or multiple other nucleotides within the mRNA at specific sites with the help of guide RNA, and with the help of some enzymes like endonuclease, ligase, they are not modifying the base, but we are inserting or deleting the nucleotides only. So two types of editing can be done. Either with the help of enzymes, we are modifying the nitrogenous base present within the mRNA. Okay, that will now code for different amino acid. Second way is by inserting certain nucleotides or deletion of certain nucleotides within the mRNA with the help of guide RNA, with the help of other enzymes like endonuclease, ligase, we can edit, we can modify the mRNA. Actually, guide RNA base pair with the specific site of the mRNA to be deleted, or to be that area to be deleted or inserted. Then with the help of enzymes like endonuclease, we cleave them. Okay, then with the help of certain polymerase enzyme, with the help of RNA dependent polymerase enzyme, we insert nucleotides like uridine. You will see them with the help of example actually. And then with the help of ligase, we join them. So in this way, in two ways, RNA editing can be done. Okay, so I hope you have understood this part. Okay, so the first part is very simple by modifying the nitrogenous bases. Second part is a little bit difficult, where we see that multiple nucleotides can be added, multiple nucleotides can be removed from the mRNA with the help of guide RNA, which are pieces of RNA that function as guide for the RNA dependent targeting enzymes or DNA dependent targeting enzymes. And they form complex with them. This guide RNA complex form a complex with them, and very often these enzymes will delete, insert, or alter the target RNA on DNA. Okay, so let us see the most common the first we have to explain with the help of an example, enzymatic modification of the bases. This is the first method to do RNA editing. Okay, so one of the most enzymatic modification of the bases occurs is the deamination of the bases. So the most common of RNA editing is deamination, which is the enzymatic modification of the nitrogenous bases, right? And most commonly, the cytosine is deaminated to form uracil, right? Adenine is deaminated to form inosine. Adenosine is deaminated to form inosine. Now, this deamination is a type of modification reaction which occurs at specific sites, or it can also occur spontaneously as a result of hydrolytic damage. So, this deamination occur at specific regions within the mRNA, or the deamination can also occur spontaneously as a result of hydrolytic damage. So due to hydrolytic damage, deamination can occur, as we have seen in your mutation chapter in your genetics. Deamination can occur at specific sites. It can also occur randomly where hydrolytic damage to the message mRNA is being occurred. In one form of the site specific deamination, a specifically targeted cytosine residue within the mRNA is converted into uridine by deamination. Now, one of the example I have said that in one of the site specific deamination, that means in particular site deamination will be carried out, there is a specifically targeted cytosine residue. That cytosine residue is deaminated to form uracil. That means cytosine will be converted into uracil by the help of the enzyme cytosine deaminase. Typically, for a given mRNA species, the process occurs only in certain tissues or cell types in a regulated manner. Now, this type of site-specific deamination, uh, um, as a result of RNA editing, due to the RNA editing, site-specific deamination occur, but it does not occur in everywhere in the tissues. In specific tissues, it occurs. In certain tissues, it occurs. In certain cell types, it occurs in a regulated manner. Okay, so it is a tissue specific process. This type of enzymatic modification of the bases for RNA editing occurs on specific tissue types. Like for example, I will give you an example based on this. Suppose the gene is expressing both of the two tissues, in the heart tissue, in the liver tissues, and also in the intestinal tissue. Okay, but in the liver tissue, due to in the liver tissue, normal editing, normal mRNA is being produced, but the same gene expressed in that intestine, that gene is slightly modified for performing a different function. Okay, it is deaminated. So I am giving you a hint actually. So we will go into this example to clarify this doubt. Okay, how in specific tissues editing is being carried out, enzymatic modification is being carried out. Let us see with the help of example, and then I will try to discuss with the help of a diagram. If I think it is given here, yes, it is given here. Okay, so the best example for RNA editing by enzymatic modification of the bases is the mammalian apolipoprotein B gene. Okay, now normally, what can I say in case of apolipoprotein B gene? Uh, if I say from the beginning, enzymatic modification of the bases with the help of this example, uh, this kind of RNA editing is found in our gastrointestinal tract system, and we know that our this gastrointestinal tract system, there is VLDL. Okay, that is very low density lipoprotein, and chylomicron particles are also present. Okay, and all of this particle VLDL, that is very low density lipoprotein, and chylomicron particles, these are all lipoproteins. Okay, lipoproteins means these proteins have lipids in them, in simple words. And one such lipoprotein is the apolipoprotein, as the name you will find out here. Okay, it is in specifically we can call this apolipoprotein B as APOB. Okay, APOB. Now the interesting thing about this mammalian APOP is that, if I say, then I will go for discuss with this lecture, that this apolipoprotein B is found in the VLDL. That means it is found in the very low density lipoprotein, and also this same apolipoprotein B is found in the chylomicron particle. So if I zoom it in to make a diagram somewhere, well, I found here. Okay, from here I can able to draw the diagram. So what I have said, I have said that VLDL, very low density lipoprotein, and chylomicron, okay, very low density lipoprotein and chylomicron particles are all what? They are all lipoproteins. They are all lipoproteins. Okay, and second, I have said that the apolipoprotein B is found in the VLDL. So within the VLDL, apolipoprotein B or APOB is found. The same apolipoprotein is also found in the chylomicron particle. But why I have write differently? because apolipoprotein B100 is protein is found on the VLDL, whereas apolipoprotein B48 is found on the chylomicron particle. Now, why so much difference? What is apolipoprotein B100 and apolipoprotein B48? It's due to the RNA editing mechanism. The same gene found within the same protein is found within the VLDL and here also, but in a different manner. Chylomicron, if I say this chylomicron is found, is secreted, it is secreted from your intestine. Chylomicron is secreted from your intestine, whereas VLDL, very low density lipoprotein, is secreted from your, yes, from the liver. Okay. Okay. Now I have said that chylomicron contains apolipoprotein B48 and VLDL consists of apolipoprotein B100. What does it mean? Apolipoprotein B48 is so called because it constitutes 48% of the sequence for apolipoprotein B100. That means this apolipoprotein B48 has 48% of the gene which codes for the apolipoprotein B. And apolipoprotein B100 refers to the gene which codes for this protein. The whole gene has been transcribed and translated to form that protein, whereas apolipoprotein 48 means 48% of the gene has been used to make this apolipoprotein B48 protein. Now the main problem, the main query of us is why this kind of difference occur in the apolipoprotein B expression on the liver and on the intestine. So I have said, if you study carefully, if you read carefully, what I am saying, if you listening carefully, you can understand that why so much of difference. That in the liver, VLDL is secreted from the liver. Okay, good. But within the VLDL, approximately apolipoprotein B content 100 apolipoprotein B100 it present, whereas in chylomicron particles which is secreted from the intestine, it consists of apolipoprotein B48. Why so much of difference? Who regulate this mechanism? That is within the chylomicron, within the intestine, 48% of the gene responsible for that apolipoprotein B has to be translated, whereas in the liver, apolipoprotein B, all the apolipoprotein B has to be, gene has to be translated. Now, in order to understand, we have to look at the pre-mRNA that codes for that apolipoprotein B. Okay, if I zoom it out now, we have to look at the pre-mRNA. Okay, we have to look at the pre-mRNA. Now, so the pre-mRNA that codes for the apolipoprotein B. Okay. Now from the pre-mRNA, pre-mRNA, your mature mRNA occurs after processing, obviously. So we have I have shown to you here that these parts are the exons which has been spliced together to form a mature mRNA molecule for that apolipoprotein B. Okay, So it has been spliced. The introns are removed out, exons are spliced. It has been shown here in this diagram. Now, this from this mature mRNA, from pre-mRNA, mature mRNA occurs after processing. Now, depending on the tissue types, depending on where this apolipoprotein B is present, on which tissue it is present, in the intestinal tissue or in the liver, such as depending upon the liver or depending upon the intestine, there would be cytidine deaminase activity. So here we see that within the liver, within the intestine, this same gene which codes for apolipoprotein B in specific tissue within the intestine. We all know in the intestine there is a specific uh, deaminase activity in the cytosine. Where in the cytosine? Within the exon 26 of cytosine. So if this is the apolipoprotein B mRNA, pre-mRNA, and this is a codon representing CAA. This codon sequence, this CAA codon is the 26th codon, although I have not drawn all the 25 codons previously. So if this is the 26th codon that is found within the exon, within this region, this C that you are going to see here, this C is going to be deaminated by the cytidine deaminase enzyme. Now, this is occurring within the intestine, not in the liver. Now, if this CAA, if this codon, which is found within the exon 26, if this CAA gets deaminated, what I have said, when cytosine get deaminated, what it forms? It forms UAA. And UAA is what? UAA is the stop codon. It will terminate the translation process, and that's why within the intestine only 48% of the gene encoding the apolipoprotein B is being produced, not all of them. So I am repeating this. So, depending on the tissue types, such as depending on the liver or depending upon the intestine, there would be a cytidine deaminase activity. Now, within the intestine, this cytidine deaminase activity is quite high and converts the cytosine into uracil. Okay, so within the codon number 2153, as I have written here, within the codon number 2153, that is within the exon number 26, within the exon number 26, cytidine deaminase activity is quite high within the intestine. It converts the cytosine into uracil. Okay, now this within the exon 26 there is a CAA sequence where this cytosine is being converted into uracil by deamination reaction. Now when CAA codon, okay, this CAA codon generally codes for what? Glutamine. Generally this CAA codon codes for glutamine, but when deamination of cytosine occur, it forms UAA now in that place. Now UAA, as we know, it is a stop codon. It is a stop codon, and thus within the intestine only 48% of the protein is going to be synthesized, because it will make a truncated polypeptide. Okay, It will make a truncated polypeptide. Now, as a result, within the intestine, there would be a translation of a partial sequence of the mRNA that give rise to a truncated protein, uh, not fully formed mature protein. Okay, so if you see this part, you can able to understood that this CAA transported to intestine site specific deamination CAA converting to UAA translation occurs only up to exon number 26, that is up to codon number 2153, and thus what happened within up to exon 26 your translation of that um, apolipoprotein B gene is being done. Okay, whereas in liver, such type of cytidine deaminase activity is absent, and thus there will be no deamination of the codon number 2153. That is, there will be no what can I say? There will be no um, RNA editing in the liver, and thus a fully mature mRNA will give rise to whole apolipoprotein B. Okay, understood? I think you have understood this part. So this is the type of RNA editing. The same RNA which codes for apolipoprotein B, uh, it is doing different functions in your intestine as well as in your liver. In the within the liver, there is no such enzymatic modification of the cytosine, and thus full protein of apolipoprotein B is formed, whereas in the intestine there is a increased activity of cytosine deaminase. Thus within the codon number 26, I think 26, yeah, 26, not codon number 26, exon number 26. If I say codon number 2153, if I say from the 5 prime end to 3 prime end, then that CAA, which is found within the exon number 26, they are deaminated to form UAA, which codes for the stop codon, and as a result, there will be translation of a partial sequence of the mRNA that give rise to a truncated protein of apolipoprotein B, which is now called as apolipoprotein B48. Okay, now why and what's the function of apolipoprotein B48? This apolipoprotein B48 within the intestine, uh, it contain only 2152 amino acids that will helps in absorption of dietary lipids from the contents of the intestine. So normally apolipoprotein B48 it helps in absorption of the lipids, dietary lipids, the lipids that we are normally using during the process of digestion from the contents of the intestine. Now, within the liver, fully functional, full protein of apolipoprotein B is formed. Where in the liver, full translation of the mRNA coding for apolipoprotein B occurs because there is no deamination reaction occurs within the liver. There is no deamination reaction occurs within the liver. Thus this apolipoprotein B100 is being formed. Um, Normally apolipoprotein B100 consists of 4563 amino acids, and this apolipoprotein B100 within the liver has a different activity. It is essential for transport of cholesterol and other lipids in the blood. So the same protein, due to truncated of apolipoprotein B within the intestine, it has a function of what? Function in absorption of dietary lipids from the contents of the intestine, whereas the apolipoprotein B100 found in the liver is essential for transport of cholesterol and other lipids in the blood. So this is the first example, this is the first explanation of the types of RNA editing. Okay. So other example of our mRNA editing of that same type, enzymatic modification, right, is your adenosine deamination. So other examples of mRNA editing by enzymatic deamination is adenosine deamination. Okay. Now adenosine deamination means we will deaminate the adenine to form inosine, right? Now adenosine deamination is carried out by specific enzymes called ADAR. ADAR full form is adenosine deaminase acting on RNA that will convert the adenine into inosine. Okay, so generally, ADAR is an enzyme, adenosine deamination, uh, adenosine deaminase acting on mRNA. This ADAR enzyme actually this ADAR enzyme actually convert adenine into inosine. Now normally this inosine is recognized as guanosine by translation, by translational and by splicing machinery, and cause um, and to change the events in the and to cause change in the amino acid sequence also. So normally, what can I say, as this inosine is recognized as guanosine due to a deamination of adenine into inosine, this inosine is recognized as guanosine. So now this inosine can base pair with cytosine, and so this change from A to I can readily alter the sequence of the protein encoded by the mRNA. So simple thing, adenosine deamination acting on RNA, this enzyme converts adenine into inosine. Now when it converts into inosine, that is from A to I, this inosine will be recognized as guanosine by the translational and by the splicing machinery, causing it potential recording events that means it actually means to say that it actually be, if the base pair with cytosine, eventually your codon sequence gets changed na. Okay, suppose AUG. Okay, no, I will not say AUG, better to omit this. Suppose AAC. Okay, now if it's A, the first A, or if the second A is getting deaminated, become AUC. Okay, now it becomes a different codon, it will code for a different amino acid. Okay, from the previous one. So I hope you have understood what I am trying to say here. Now this type of it has been said here that this type of RNA editing, that is cytosine deamine not cytosine deaminase, ADAR enzyme, where it deaminate the adenine to form inosine, this type of mRNA modification is done in specific regions. So an ion channel expressed in mammalian brains is the target for this type of editing. So where it occurs? This A to I editing is prominent within the brain specific transcripts. An ion channel expressed within the mammalian brains is the main target for this type of A to I editing. A single edit in its mRNA causes a single amino acid change in the protein, which in turn altered the calcium permeability of the channel. In absence of this editing, brain development is seriously impaired. So this type of editing, the first type of editing that we have seen from C to U, this type of editing we have found in the within the apolipoprotein B. in the liver and in the intestine, right? But here here in case of A to I editing, that is from adenosine to inosine, this type of editing occurs within the mammalian brain, okay. The an ion channel expressed in the mammalian brain, that means the within the mammalian brain there is a ion channel. That ion channel is actually is subjected to editing, okay. Now within that, the gene responsible for that ion channel formation, it causes a change in the its adenine into inosine at certain site, which causes change in the function also of that ion channel. It alters the calcium permeability of the channel, okay. That means this channel is specific for the entering of certain ions, okay. Here the calcium permeability alters if there is slight change in the gene responsible for the channel formation, okay. And it has been said here that if this type of editing does not occur in the brain, then it cause serious it will seriously um, affect the brain development actually. So I hope you have understood this part what I am trying to say. I am again trying to say in a simple word. A to I editing, that means from adenine to inosine editing, is done by a enzyme adenosine deaminase acting on RNA. Okay. Next, where this type of editing occur? This type of editing occurs within the ion channel found in the brain, found in the mammalian brain. That means the gene responsible for that ion channel within the mammalian brain is subjected to this type of RNA editing, A to I deamination editing. Now, due to deamination from A to I, a single amino acid change occur, which actually changes the function of that ion channel. It alters the calcium permeability of the channel, and it has been observed also that in the absence of editing, if this type of editing does not occur within the mammalian brain, if this type of editing does not occur in that channel protein, then brain development is seriously affected, it is seriously impaired. So I hope you have understood what I have said about this type of RNA editing. The next mechanism of RNA editing is insertion deletion editing. That means insertion of the nucleotides, deletion of the nucleotides. Now I will discuss with the help of an example actually. Otherwise it will be problematic for you. Now, what I have said that in the second type of RNA editing, which is your insertion, it is also called as insertion or deletion editing. Now, this type of editing occurs as a result of insertion or deletion of multiple nucleotides with the help of guide RNA and with the help of other enzymes. Here we are not modifying the already present bases found in the mRNA. We are uh, inserting multiple nucleotides, such as we are inserting multiple uridines, okay, or deletion of certain nucleotides within the mRNA molecule, and also with the help of guide RNA and with the help of enzymes. And what I have said about guide RNA, that guide RNA is a piece of RNA that function as to guide for the RNA or DNA targeting enzymes. So the RNA or DNA targeting enzymes, they are allowed to interact with the mRNA in order for insertion and deletion of a certain sequence within the mRNA with the help of guide RNA. They form complex with the guide RNA, okay. And very often these enzymes like endonuclease, ligase, okay, other enzymes are also there. You will see, the name. They will delete, insert the target RNA. Okay, actually, what happened? The guide RNA base pair, the guide RNA base pair with the mRNA at specific site where RNA is to be edited, and then that guide RNA will serve as a template for addition or removal of nucleotides within the target RNA. Again, I repeat, here actually the guide RNA will base pair with the target mRNA at a particular site within the mRNA where RNA has to be edited. And then that guide RNA will serve as a template for addition or removal of the nucleotides within the target RNA. Such type of RNA editing is found within the trypanosomes. Now, what is trypanosome? Trypanosome, what can I say? They are unicellular parasitic protozoa that belongs to the Trypanosoma genus. A large number of species of trypanosomes and subspecies of trypanosomes have been identified and has been studied extensively. Okay, and these are flagellated protozoans. Okay, they have a flagellar tail. Okay, and they have a dense granule of DNA within their mitochondria. So what we are trying to talk about the mitochondria of the trypanosomes. Okay, within the mitochondria of trypanosome species, this type of editing occurs. Okay, now many things has to be mentioned here. uh, Three terms I want to mention. One more a term I want to mention is your what is trypanosomes. I have already said that trypanosomes are flagellated protozoans. They have a flagellar tail. Flagellated protozoans, and they have a they have a large mitochondria. They have a large mitochondria, and within the mitochondria there is a kinetoplast. This is the term that I want to explain with you. They consist of a kinetoplast. Kinetoplast is what? It is a network of circular DNA, also called as kDNA, kDNA, that is found within the large mitochondria that contains many copies of the mitochondrial genome. So it looks like a granule, okay, a dense granule of DNA found within the mitochondria. Dense granule found within the mitochondria of kinetoplast. Now, within this dense granule, it consists of many copies of your mitochondrial genome. Okay, and what is the specificity of this trypanosomes? That there is a specific species of trypanosomes called as Trypanosoma brucei. Brucei, Brucei. Trypanosoma brucei is actually a parasite which cause African trypanosomia, which is called African trypanosomiasis, which is also called as African sleeping sickness, and it is done with the help of a vector, carrier, a fly known as TCTC fly, which is a sand fly, type of biting flies actually, which transmit this Trypanosoma brucei to the host to form this type of African sleeping sickness. So while studying them, um, about the trypanosomiasis, they identified that within the trypanosomes they have a large mitochondria, within them they have a large granule, and scientists have curious to find out what contents within the granule, and they name them as kinetoplast, a type of um, to I they actually they want to they observe that if they contain DNA, that means it contain information just like a nucleus. So that's why they have named them kinetoplast. And by observing them, they found a large number copies of mitochondrial genome is being found, and within that site also a different type of RNA editing is also being done, which is your insertion and deletion editing. So let us see how it is being carried out. So it is saying that in the trypanosomes, a different form of RNA editing is carried out within the mitochondria. In this case, multiple uracils are inserted, or uridines you can say, are inserted into specific regions of mRNA. So within the mRNA, the mRNA within the mitochondrial mRNA, there are specific regions where multiple uridines has to be inserted after transcription, or in other case U can also be deleted. Okay, the multiple U's can also be deleted. In addition, the addition of U to the message change the codons. Obviously, if you are adding uridines multiple times within the mRNA, certain mRNA in the target site, then obviously it will shift the codons, it will change the codon sequences and reading frames, completely altered the meaning of the message RNA. Okay, so it will completely change the meaning of the messenger RNA after multiple U is being added. Example of such type of RNA editing is found within the trypanosome COX2 gene. Okay, so in the COX2 gene of trypanosome, this type of insertion deletion editing occurs where multiple uracils, multiple uridines are being added to certain site of the mRNA. Thereby, it causes change in the codons, change in the open reading frames, and completely altering the meaning of the mRNA. I hope you have understood this part. So, what it is saying that let us try to explain this actually in a practical manner. Within the COX2 mRNA of the gene, there is a specific region where four uridines can be inserted between the adjacent bases at three sites. So within the COX2 mRNA of the gene, there is a specific region where 4U or 4 uridines can be inserted between the adjacent base at three sites. 2 U's at one site and 1 U each of the two additional sites. So four regions has been mentioned. With the help of diagram, you can able to see that uh, within the three sites, four uridines can be added within the COX2 mRNA. In addition, this addition of U can alter the codons and cause change in the reading frame. Obviously, that is required to generate the correct open reading frame. So because if you are adding uridine at certain sites, in whatever sites you are adding, it will ultimately change the open reading frame. It will ultimately change the codons also. Okay, now how are these additional bases inserted perfectly to that specific sites? That's the question. That okay, we are inserting certain nucleotides to change the coding sequence, to change the exon, to change the open reading frame. But how these additional bases are perfectly added to that site only? Now it can be added to that perfectly to that site where they want to edit that site with the help of guide RNA actually. So U nucleotides or uridine nucleotides are inserted within the with the help of guide RNA. Well, guide RNA ranges from 40 to 80 nucleotides in length and has three specific regions. Guide RNA helps the other enzymes to interact with the mRNA and cause that insertion deletion of the sequence also. But specifically, if I say about guide RNA has three specific regions: an anchor region at the 5' end. Within the anchor region, it will direct the guide RNA to the region of mRNA where it will edit. That means there it will actually bind base pair with the mRNA actually, and it is also the site where guide RNA will be where the editing of the mRNA will be done. It is called the anchor region. Anchor means to hold on. Then comes the editing region. Editing region is located just after the anchor region. You will see in the diagram the editing regions which is located just after the anchor region. Now within this editing region, it will determine exactly where the U will be inserted within the edited sequence. If I talk about the mitochondrial genome of trypanosomes, then I have to, with the help of example, we have to say this, that editing region will determine exactly that where this uridine will be inserted within the edited sequence. And lastly, within the guide RNA, there is a poly U stretch also, multiple poly uridine residues at the 3' end. So guide RNA have three important regions. One is your anchor region, one is your editing region, and another is the poly U stretch. Anchor region actually hold the guide RNA with the mRNA at specific site to be edited. Editing region will determine that exactly where the U has to be inserted within the edited sequence of mRNA, and poly U stretch is the multiple uridine residues found within the guide RNA. Okay. Okay. So let us see about the different regions. Let us see the diagram first, then I can able to discuss with you. So here you can see the mRNA shown in green and your guide RNA shown in yellow. Okay, so here is the anchor region, the region of homology where they are base pairing with one another to hold the mRNA in that site to be edited. Then there is a editing region. Well, editing region editing region is the region where your editing will be done, where we will edit that site by inserting, here we are inserting multiple uridines. Okay, and lastly there is a poly U tract also where multiple uridine residues are being found within the guide RNA. So in this way, guide RNA base pair binds with the mRNA to be edited. Okay. So let us see about the anchor sequence of the guide RNA. The anchor region of the guide RNA consists of a sequence that will base pair with the region of an mRNA immediately besides to the region that will be edited. That means the anchor region actually, what can I say, within the anchor region, the guide RNA actually base pair with the mRNA, and this anchor region is located just immediately, if I say from the 3' end to the region, it will be edited. That means beside the anchor region there is a editing region, as I have mentioned you before. Now this is followed by editing instructions also. It is followed by editing instruction also. A stretch of guide RNA complementary to the region of the mRNA to be edited, but containing additional adenine. That means there is a RNA within the editing region of the guide RNA here in this example of trypanosoma mitochondrial DNA. The editing region here consists of complementary regions, okay, but it also contain additional adenines. These adenines are at the positions of the guide RNA opposite where U will has to be inserted to the mRNA. So within the guide RNA editing region, there is additional adenine regions as well. And these additional adenines will be used to complementary base pair with uracil, where we will add within the mRNA. So I hope you have understood what I am trying to say. Let us say again. So you can see that this is your anchor region where it base pair with the guide mRNA. The editing region you see. Now if you focus on the editing region, it also consists of what? It consists of adenine residues also. Now these adenine residues will be used uh, for guiding the instructions that where U has to be added within the mRNA. Okay, I hope you understood what I have tried to say. So that's why it is saying that it consists of additional adenines. These adenines will be at the positions within the guide RNA opposite where the U will be inserted into the mRNA. Okay. So these adenines are found within the editing portions of the guide RNA opposite where U has to be inserted. Where uracil has to be inserted within the mRNA. The guide RNA and the mRNA forms an RNA-RNA duplex with which will loop out a single-stranded regions opposite where U will be inserted. Usually, because the editing regions within the guide RNA is not similar. Okay, it has multiple adenine residues here in this example, and that's why what happened within that site complementary to the mRNA complementary to the guide RNA is the mRNA where uridine has to be inserted. Okay, they will looped out actually. Okay, they will looped out. They form a loop like structure where adenine is present in the guide RNA. So they are base pairing with the mRNA. The guide RNA and the mRNA are base pairing with one another. That's good. They are base pairing with one another. They form an RNA-RNA duplex, which will loop out single-stranded regions opposite where U has to be inserted. So there is looping formation occurs if you see it carefully. So now the guide RNA and the mRNA are base pairing with one another, but where the adenine residues are there, uh, now complementary to these adenine residues, we have to insert U. We have to insert uracils. Okay, to be edited actually here in this example of trypanosomes mitochondrial DNA, mitochondrial RNA. So that's why a loop like formation occurs here. Okay, now it will be identified by endonuclease enzyme to cleave at this site where they will insert the they will they cut the site, and within the site complementary to the adenine they will place what? Uridine. Obviously U will be added as guided by base pairing with the guide RNA and editing region. This region will be used to add uracil here because adenine is there. So here it is saying now. So the guide RNA and mRNA forms a duplex RNA duplex, and where the guide RNA editing region has multiple adenine residues, it will looped out actually at that time. Then an endonuclease enzyme will recognize that loop region. The endonuclease will recognize this loop region as what? Mismatch basis. Okay, that's why complementary to that loop regions where guide RNA is base pairing with the mRNA, that mRNA is cleaved on that site. Okay, by the endonuclease enzyme and cuts the mRNA opposite to these loops. Okay, so the opposite to these loops, endonuclease enzyme cleave. Okay, because it identifies as a mismatch. Now, editing involves transfer of use, that means uracils to the gap in the mRNA. So within the mRNA, editing is done. Multiple uridine is being added. Insertion of U to the mRNA is catalyzed by an enzyme called 3' terminal uridilyl transferase. Now, this transferring of U, so after this loop formation in the editing region of the guide RNA, endonuclease identify them as a mismatch, so it cleaves. Okay, now in that site multiple, so in place of A place of a complementary base pairing of U occurs. So what happened? Uracil is being added. Now this is catalyzed by enzyme called tutase. What is tutase? Tutase full from? Tutase full from is 3' terminal uridilyl transferase. 3' terminal uridilyl transferase or tutase. It actually catalyzes the addition of U in the gap in the mRNA, which has just been cleaved by the endonuclease enzyme. Now, after addition of multiple U, the two halves of the mRNA are joined by RNA ligase, and the editing region of gRNA continues its action along the mRNA in a 3' to 5' direction. So after action of these two, after the addition of the uridines, the two halves of the mRNA are joined by an RNA ligase enzyme, and the editing region of guide RNA continues its action along with the mRNA. It will now move to a next site where it has to be edited in a 3' to 5' manner, not 5' to 3'. So it is moving from 3' to 5'. That's this guide RNA, and it is again searching for a site where editing of the mRNA will be done. Again it will bind. Then at the editing region there is multiple, in this example there is multiple adenines, and thus a loop formation will occur because it will not base pair at that regions. Okay, and this will be recognized by the endonuclease enzyme, which will cleave them, and in that site with the help of tutase enzyme, that means 3' terminal uridilyl transferase will add uridines in that place and will be sealed by ligase again. So this will continue, and in this way editing will be done. So normally a single guide RNA is responsible for inserting several uridines at different sites in a 3' to 5' direction. So from 3' to 5' direction, a single guide RNA is responsible for adding of uridines. Furthermore, in some cases, several different guide RNAs work on different regions on the same mRNA. Okay, so here we can see in this diagram that first I have said that the guide RNA is base pairing with the mRNA. That's good. There is the anchor region where complementary base pairing of the guide RNA and mRNA occurs. Then we see an editing regions where some adenine residues are present, which is not complementary to the mRNA, and these regions will actually specify that where U has to be added, urine has to be added, and due to this region of complementary base pairing, they will form a loop like formation because here mismatch occur na. No base pairing occurs between the mRNA. This will be recognized by endonuclease and cleave at that site, and after cleaving, then tutase enzyme 3' terminal uridilyl transferase will catalyze the addition of adding U in this site, and as you can see, U is being added here. U is being added here as guided by the guide RNA and editing region, and then ligase will join the gap. Ligase will join the gap, and this will continue. It will continue to move in this fashion from 3' to 5' manner, because respect to this mRNA, it is moving from 3' to 5' direction. The guide RNA is moving. Sometimes multiple guide RNA is also involved in RNA editing. So I hope you have understood what I have said up to this much. If you have any doubts regarding this RNA editing process, you can ask me again. Okay, I will give you a separate video link, which is a very good animated video, as mentioned um, by in the YouTube. So let's go through that video also, and that's all for today's lecture.